All right, so welcome everybody to this fifth lecture of the Virtual Brain and Clinical Research course, an introduction. So today we're gonna speak a lot about the fundamentals of the virtual brain, that is the theory behind it, its main components, its main uses, objectives. So there's gonna be quite some theory, a little bit also an excursion into dynamical systems theory. However, all of this I plan to do in a way that not much expert knowledge in mathematics or programming would be needed. So first of all, I hope, let me see how can I advance. Yep, so I guess you already know about the website of the virtual brain, where you can find all relevant information. It's very comprehensive. You'll find tutorials, the software itself, news, all the nice research done using the virtual brain. And I have to thank all of my colleagues in the group that helped me compose this talk. You see some of them here, together with their virtual brains. Especially, I want to thank Misha, the one here on the left, and Professor Drs. Petra Ritter and Victor Irsa for providing me a lot of the materials for the slides you're going to see today. So first of all, let's start with what is TVB and why is needed. So responding to this question in one phrase, we would say that TVB is indispensable in linking brain anatomical and neuroimaging data to unravel the secrets of brain function. Now, what does that mean exactly? So during the last decade, neuroscience has advanced a lot, especially in terms of collecting all kinds of data, from neuroimaging, structural, functional MRI, EEG, MEG, optical imaging. We're gathering tons of data. But we might say that maybe the understanding of how brain functions has not advanced equally. There seems to be a need for a way to compress all this data and represent it in a way that can help us advance theory, the understanding of the brain, as well as make the translations to clinic. So this is exactly where the need for a model or where a model of the brain can be useful, as long as it helps us compress all this data, or let's say compress the data and our theoretical knowledge and assumptions, test hypotheses, make predictions, etc. More concretely, as I said, there has been quite some progress in non-invasive brain imaging in the 90s. This led to a boost to the network science application in the brain. There was a lot of large-scale brain network modeling, the use of graph theoretical analysis, whereas the Human Connectome project has made a lot of effort to represent all the connectivity of the brain. Later on came the rise in computational power that allowed the emergence of technologies like the virtual brain. And lately, this has led to nice ideas about clinical applications. You will see later on some instances of the science behind the clinical trial of Epinov in France, which is about helping the treatment of epilepsy through surgeries. There are other initiatives like the Virtual Brain Cloud, where I'm working, that tries to bring data and the virtual brain together from different sites all over Europe. So there is a lot of it going on, but what I would like you to observe is that it was first that there was a lot of progress concerning mainly the structure of the brain, especially the structure as a network, while the latest part that relates more to TVB has also to do with the function of the brain. And this is exactly, these two words, structure and function, is exactly where TVB stands, and we're going to use them to understand more about it. So we would say that the main, in one phrase, the virtual brain is a model that is useful for linking the brain structure and function. Now, I'm going to go through an overview of the virtual brain within a few slides, and then we're going to go deeper into each one of its components. So first of all, there is a lot of theory behind the virtual brain, and there are assumptions that come from theory about first of all about cortical homogeneity. The idea is that the cortical sheet is relatively homogeneous. It is composed of cortical columns, as we know, and within these cortical columns, we find the neural networks. Those neural networks can be represented at different scales. We can have, as we know, detailed biophysical models of neurons. Then we can start abstracting from them and treat them as point neurons that can fire spikes. And then we can also have statistical descriptions of populations of such neurons and represent their activity as, for instance, firing rate. Now, the second important assumption is how exactly this function is reflected in brain measures, like spikes, local field potentials, EEG, MEG, fMRI, etc. So one thing, therefore, is assumptions about the forward modeling, about how function, in that sense, the activity of neurons within the cortical seat, how it translates to the observations we have from the brain. It is exactly at this scale of transferring or expressing this function into brain measures where TVB stands. Yeah, as I mentioned, we know now, and we can have quite accurate forward models for each one of these measures that take into consideration the geometry of the cortical seat, as well as anything else that exists between the measurement and the activity of the neurons at different scales. Now, when we speak about function, though, again, we stand at a specific level of detail. We represent brain function mainly as oscillations in a large-scale brain networks. To do this, you can see here evidence from different publications where the mean field activity of many neurons, for instance, here expressed as spike trains, can be summarized as local field potentials, and there is evidence that there is a very high correlation between the LFT gamma power and spike trains. So this gives us the idea that we can actually model large-scale oscillations in the whole brain, the things usually we measure with fMRI, EEG, etc., as the activity of interacting neural populations, each of which is described by its mean field. In other words, by its average behavior. For instance, again, here in another instance of a model we're going to see later on, we see a population of neurons spiking and the mean activity, which is like a smooth curve. So this is exactly the level of description that gives rise to the observations we have from the brain, and therefore this is the computational unit of TVB. So bringing all these things together, what we have is, on the one hand, the geometry of the brain, the cortical seat, which is very important for our forward models for measurement. We have a homogeneous cortical sheet, which means also possibly homogeneous local connectivity around its neuron. The computational unit of the neural population, which becomes a node in our network. And finally, long-range coupling between brain regions through white matter tracts, which gives us the heterogeneity in the network. So these are all together the main model components of TPP. This is a first demonstration of what we mean by function, by large-scale network function of the brain. You see a video demonstration, an animation from a study that we're going to speak about also later on by Sirnan et al. This is a whole virtual brain model that is tuned against fMRI data in a way that manages to reproduce a series of observable phenomena of neuroimaging, like the scaling of EEG source alpha power, the anti-correlation between alpha power and fMRI, and the main fMRI resting state networks that we detect when we do a correlation analysis of fMRI. So this is the kind of science we want to do. We want to have a brain model that we can simulate and test against neuroimaging data that we have available. Okay, I just leave here 3 main basic references from TPP where you can find all of this theory and mathematics and models and assumptions. So you can go and look more for yourselves. And now let's try to look deeper into the separate components. Let's start from the virtual brain structure, specifically the topology and connectivity of the brain. Let's go first through visualization where we see these different components. We have the skull. If we look inside, we have the homogeneous cortical seat. And if we look deeper inside, we have the white matter tracts that connect distant brain regions. And now it is exactly this cortical sheet that we can split into different brain regions following brain atlases, which is called a parcellation. And then we can go even more abstract and shrink each of these brain regions to its center of gravity and form a network, the links of which depend on the white matter tracts you saw earlier. So this is the highest level of abstraction we can go with the large-scale network model of the brain, where we finally represent it as a network, or where each node is a neural population and it's linked and interacts with other nodes through the white matter tracts. Come on. Okay. So first of all, as I've said, the first element of its structure is the cortical sheet geometry and parcellation. We get MRI, structural MRI measurements, and we can reproduce the surface of the brain and represent it as a triangulated mass. As you see here, there are nodes and small triangles, and they follow the geometry of the cortical seat. And then using specialized software like FreeSurfer, we can actually, and publicly available atlases of the brain, we can actually attribute each one of these patches of surface, the cortical surface, to different brain regions. There are several atlases out there, some of them based mostly on structural differences of the brain regions. Some others also take into consideration functional elements. Now, based on this topography, we can have at least 2 ways, or actually 2 ways to model the brain. One is the case where we're actually shrinking each one of these brain regions to its center of gravity, and we form a network of nodes that have no local structure. This is what we call the region-based modeling, and we can do this and have in the end something like everything between 30 and 1,000 nodes. Or alternatively, we can simulate, and we can model and simulate along the whole surface using every vertex of these triangles, of the triangular mass, as a separate node of the network, in which case we can also have local interactions. In this case, obviously, we can have much more detail, and we can have simulations with several tens of thousands of nodes. Now, we need also to connect this network, of course, and this happens through global and local connections. First of all, global connections, which we construct via diffusion tensor imaging. We start from diffusion MRT, and with which we can track axon bundles as they leave one region and go to another. There are algorithms that help us reconstruct the diffusion directions, and therefore then using deterministic or stochastic algorithms, we can actually get representations of the white matter tracts, and we can count them and we can measure the lengths. And like this, we can represent the brain as a network where different brain regions are the different nodes, and these are connected through connections that have a weight, which depends, it's some transformation of the counts, how many tracks we find between every different pair of nodes. And also they have a length, and this is very important because this length can be used to determine the time delays of communication from one brain region to another. When we don't have this, we can also use, of course, just the Euclidean distance in 3D. And this is quite important because both of them, weights and time lengths or delays, create a space-time structure for the connectivity of the brain network, which is represented here in a way where as we increase time delay, we find different connections on every being, let's say, of this histogram of time. So when we look at the shorter connection, we have some of them, and when we look at longer, we have some others. And this is what we would say that shapes the music of the brain. Think of it as a kind of chord instrument, for instance. When we play the guitar, for instance, we hit the chord, and depending on the length of the chord, we get a different musical tone. It is a bit like that for the brain dynamics. It's not only the weight, the strength of the connection between two regions, but also the distance, the time delay for communication that plays a very important role for the resulting global dynamics of the brain. Now, coming to local connectivity, remember that at the local level, we have the assumption of homogeneity. So first of all, this applies only when we do a surface-based modeling. And local connectivity refers to connectivity that depends only on the distance between the nodes in this triangular maze. And usually it is the geodesic distance, so the distance along the surface. For the local coupling, usually we define local connectivity spatial kernels. These are functions that only depend from on distance, and there is no time delay. So they look usually a bit like that. So if we have a node here, it will be coupled to all nodes in its neighborhood, and the strength of the connection depends only on the distance. So wherever we go on the brain, the coupling is homogeneous. It only depends on the distance. Now I'm going to show you a little bit what we said earlier, that about space-time structure and how important this is. Following this and several other studies with a similar subject, I'm going to show you a demonstration. Here we model the brain surface as a neural field, as a continuous cortical seat, so that the dynamics of it, the oscillatory dynamics of it, have a spatial and a temporal component. It's modeled as partial differential equations. And what happens is that there is homogeneous local connectivity in its neighborhood of this cortical seat, and heterogeneous large distance connectivity. And you will see how changing the distance of this connectivity, and therefore changing the time delay, actually changes the oscillatory patterns we get along this space. So we get different oscillations for a small distance, different ones for a larger distance, and even more different ones for an even larger distance. We can look at this as a video where we gradually change the parameter of the distance between the connections of the local networks. And this destabilizes the previous kind of dynamics and eventually take us to a transient, to to a different mode of a different spatiotemporal pattern along the neural sheet. Yeah. So distance matters, times matters, and therefore this space-time structure of brain connectivity is a very crucial element of the whole large-scale network modeling of TVB. Okay. Now, let's get deeper into what we call function in the brain, in the virtual brain. This function is described as large-scale dynamics that are derived from connected population dynamics. And I present you here with the most general equation of TVB. So it comprises of 4 terms in this equation. The first term, and actually this is the equation that governs the dynamics of a computational unit of TVB. I remind you, the computational unit might be a whole brain region if we are doing brain region modeling and simulation, or it can be even just a small vertex in this triangulated surface. Actually, the way this equation is formulated using integrals, it actually points to a surface simulation. So there are the following terms. There is, first of all, some dynamics that define the spatial dynamics of each neuronal population, the way it oscillates or whatever else. Then there is a part of the local homogeneous connectivity. You see that it only depends on the distance between our point X and every other point X prime on this surface. Then there is the global heterogeneous connectivity. We know it is heterogeneous because it depends on the 2 points that are connected, and this can be anything. And this connectivity also has a time delay. This time delay depends on the distance, on the geodesic distance on the surface, divided by the speed of communication. And finally, we can have an extra term of noise that are stochastic additions to the whole dynamics. In other words, dynamics that are not explained by our equation. It can be a Gaussian white noise, or it can be a colored noise that has time correlations as well. What is important, though, is that this is really the computational unit of the TVB, the neuronal population. Exactly, and this is where the equations of it lie. We can simplify this equation for brain region modeling. We don't have any more integrals, we have sums. And this is a very simplified version of the same equation. Still, we have the same kind of equation for the population model. Here we have there is no local coupling, obviously, and the global coupling here has a very simple, it's a very simple linear function where inputs coming from other nodes are just weighted by the weights of the connectivity, and they arrive with a time delay based on the delay matrix. And finally, there is the noise term. So usually, in most cases, this is the way our model looks like. Now let's go deeper into this local population dynamics, the computational unit, let's say, the dynamical unit of TVB. What is a population dynamics? Let's take an example from nature. You see in this video that there is a population of birds in this case, and they fly around, and we know that they form these nice forms exactly because they have local interactions among them. In some way, they're following their neighbors, let's say. So they have local interactions. These interactions are generally homogeneous. They only depend on distance. And therefore, although there's a thousand birds there, it's likely that we can describe these 3D dynamics of their flying with much less equations, much less degrees of freedom. We can summarize these forms with a very simple model, most likely. or anyway, much simpler than the 1,000 degrees of freedom if we had to really model every single bird flying. Now imagine that we model the brain, the large-scale brain dynamics as populations of neurons like that are additionally connected through these heterogeneous long-range connections. So it's an even more complex situation, obviously, in the brain because we have several thousands of those. But here I want to pass you the message that we're going to do a similar thing with neurons and try to have population dynamics described through a very few, very simple equations of oscillators. Okay. So in TVB, we have several population models that we can use. There are models where the main state variable that describes them, it is something like a local peak potential or synaptic activity. Others, they model something like the firing rate activity of neurons. Then there are models that are purely phenomenological. In other words, they model only the kind of dynamics like oscillatory or something else without being able really to identify their state variables with a biophysical quantity. What we don't have, though, is more cellular models. We don't represent neurons here. We only represent populations of them. In all cases, all these models, what have in common is to be described by nonlinear dynamic systems via differential equations. So that's why here we can make a small pause, open a big parenthesis, and go through a small tutorial on dynamical systems so that we understand better the way that the function is represented in TVB. So what is dynamics? Dynamics is a way to describe change over time. Here, for instance, we have a variable that we record in time. It could be the potential, for instance, or the firing rate. And what we have below is actually the same phenomenon described as the rate of change of this potential, the derivative. So if this is the way V changes in time, we represent this change through the derivative. This allows us to have a different representation that doesn't depend on time explicitly. For instance, let's start from the beginning of the movement here, and let's try to represent it in a space that is spanned on the horizontal axis by the potential, and on the, well, it seems to be the other way around actually, but we'll find out. It is no. Yeah, so on the horizontal axis, in this case, we have the derivative, and the vertical axis, we have the potential. And then we would represent this movement like a spiral. Okay, so imagine we are at this point, which is here, and we perform a damped oscillation, which means we are spiraling towards the final equilibrium point. So here we can see several elements. First of all, this is what we call the state space, a space where we can represent the trajectory of the phenomenon and also represent have a very explicit and deterministic representation of the way the movement will evolve in time, although there is no explicit representation of time here. And we have the initial condition, and we have also the equilibrium towards which the movement evolves. In another case, we can have an oscillation, for instance, a steady state or self-sustained oscillation. Similarly, in this case, we have a similar representation in the state space, again, having a state variable and its rate of change. And such a a steady state oscillation is represented by a so-called limit cycle, which is a closed trajectory. Wherever we start from, we will eventually end up on this circular dynamical structure. Now, in a similar way, when we go to more further dimensions, we can also have more complex dynamical structures. So it's like a little bit like a statue, right? It's like taking a dynamical phenomenon that evolves in time and represent it through a static dynamical structure in a relevant state space. So we can have closed trajectories, oscillations, or we can have spirals like this. And this reminds you, of course, of neural bursts. And we can also have chaotic dynamics where they are bounded in a space. However, they never really cross from the same point twice. And what is more interesting now is that when we change some parameters of our equations, of our differential equations that describe the dynamics, we can actually have qualitative changes from this and have the one dynamical structure pass into another. For instance, from a spiral that we saw at the beginning to a sustained oscillation or to chaotic dynamics. And this is what we call a bifurcation. So bifurcation is when we change the parameter of the dynamics, and this leads us to a different, qualitatively different dynamics. And we also, this is also what we call the state space, sometimes as phase space. It comes from the phenomenon of changing the phase of the water from ice to vapor to liquid water when we change parameters like temperature and pressure. So it's a similar situation here, and historically it has evolved as to call the state space also as phase space. So on the one hand, we have the representation of dynamical function through dynamics in the state space. On the other hand, we have parameters that when we change them, we can pass from one structure to the other, from one qualitative dynamics to the other. Let's see how this works for dynamics of 1 and 2 variables. First, for dynamics of 1 variables, like the ones we saw earlier with the potential. So we have these different kinds of representation of the dynamics. We can have trajectories in a relevant state space. We have time series described as the evolution of an amplitude or whatever else along time. And we can also have phase portraits. In the first representation, we see how a system that has a stable equilibrium looks. Wherever we start from, we follow down the potential curve to a stable point. Or when it is unstable, the opposite happens. There is a so-called source of activity here, and the slightest perturbation will take us away from it, either from the one or the other side. Or if it is just like that, then it's indifferent and every point is equally stable. This is represented in time either as an asymptotic trajectory towards an equilibrium point, or as staying where you are or where you're put, or in the case of unstable dynamics, asymptotically leaving from the equilibrium point. The corresponding representation in the phase space, remember, we only have one variable, so the phase space is actually a line. And in this line, a stable point is symbolized like here, a filled dark dot, which attracts the flow from both directions, whereas an unstable point is symbolized as an empty circle that sends the flow away anyway, repels, sorry, that was the word, repels the flow away from it. Let's see an example now. So imagine we have a flow with a stable fixed point, a stable equilibrium point, in other words, and then an unstable equilibrium point. So give me a second. the closer we are to this equilibrium point, the slower the flow, because equilibrium point is actually the point where the derivative of the system is 0. The same is also for here. An unstable fixed point also has a derivative of 0. But the difference is that the stable fixed point will attract the flow, while the unstable point repels it. This we can see if we add an axis here to represent the derivative. Then here we are at the point where derivative is 0. And as we go further from the stable fixed point, the derivative becomes higher and positive on this side, higher and negative on this side. So positive means that the flow will go to increasing values of x, oh come on, while negative means that we will go to lower values of x. So the stable point attracts the flow, and the further we are, the faster we go. The unstable fixed point repels the flow, and the further we are from it, the faster we're going. All this we can see here also. Now we see the same things we saw earlier. So we have the flow on the line, the stable and unstable points, which correspond to the point where the derivative is 0. And we can see now an example of what we call a bifurcation or a qualitative change in this dynamics. So initially we have this system where, given that the derivative curve does not ever cross the point that is 0, this is a system without any equilibrium point, and wherever we are on this line of fix, we will keep increasing infinitely. And the further we are actually from this area, the faster we will be going. So actually what we're going to have is fast, 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 slow down, fast, 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 going away to the infinity. However, if we change a little bit this parameter, oh, come on, and make it go a bit down, then we're going to actually reach and touch the point where the derivative is 0. In this case, we get what is called a saddle. A saddle is a point that attracts the flow from one side and repels it from another side. So what we would have is fast, 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 slowing down to 0. And unless a perturbation externally arrives, we're going to stay here forever. But this is, if a small perturbation pushes us a little bit on the left, we're going to still go back to the equilibrium point. But if a small perturbation actually gets us to increasing x values, then we're going to leave and progressively speed up and then go again far to infinity. However, and this you can see also in time here, okay? This is the way we approach, and this is the way we leave the saddle. And then if we change a little bit the parameter k, then we will actually take another qualitative change to the system and lead to a point like this, what we were seeing earlier. We have a stable fixed point and an unstable fixed point. This attracts the flow from the one side and also from the other. The further we are, the faster we go. The other one instead repels the flow. So if someone starts around here, he's going to leave slowly, then speed up, and then slowly again reach the stable equilibrium point. However, if someone is here, he will leave and go again far to infinity. So this changes from qualitative changes that this change of parameter brings is what we call a bifurcation. And on the line, on one-dimensional flows with only one dynamical state variable, this is all we can get. We can get a series of fixed points that alternate between stable and unstable. However, when we go to two-dimensional flows, which is by the way more realistic because we need at least two state variables in order to have oscillations. For instance, we have here an example of some equations. Here, x and y can be considered as transformation of position and velocity. And we have the derivatives, the way position changes and the position of velocity changes. We have here one kind of dynamics of spiraling towards an equilibrium point. And we can, as represented in time, we have x in blue and y in red. and the way this is represented in the state space. What we can do now on this state space is we can compute these equations for every possible combination of x and y, and therefore we can get a vector at each one of these points that points towards the way the flow of the system will go. So if I leave you here, you're going to travel along this way and end up on this equilibrium point. If I leave you here, you're going to do a similar movement, etc. So this vector field here represents actually the flow of the dynamics. Additionally, though, we can define the following objects. We can define the lines where the x derivative is 0 and the y derivative is 0, separately one from the other. So these are represented, these are the so-called null lines, and they're represented here. For instance, the line x is 0 is this line, the y-axis. This line here is this one. And for the y derivative, we have the line y is 0, which is the x-axis, and x is 1, which is actually this one. And this, I said again, are derived by just solving these equations by setting them equal to 0. Now, where the null lines cross is where we have equilibrium points, because both of these are 0, right? The derivative of both state variables is 0. In that way, we get 3 equilibrium points, one here, 0, 0, one here, 1, 1, and one here, 2, 0. And we represent them here also in the vector field. So this actually, the two of them are saddles, because you see they attract the flow from one direction and repel it from another. They attract the flow from one direction, they repel it from the other. Whereas this is a stable equilibrium point that attracts the flow, and the flow arrives to this point asymptotically. So we see that there are more things that can happen in 2 dimensions when we have 2 degrees of freedom, 2 state variables. Now, bifurcations can also obviously happen here. I just show you a picture of an example. We have a similar set of equations of 2 dynamical variables. And depending on the parameters here, we can get at least 3 different dynamical objects. We can have a limit cycle, which, as we said, is a closed trajectory, and it models oscillatory dynamics. So you can see the vector field, and it has an unstable point here, which is causing the limit cycle to emerge. So if we start from here, we're going to end up on the limit cycle and oscillate forever. If we start from here, asymptotically, in all cases, we go to the limit cycle behavior. If we change a little bit the parameters, though, we have a different structure with a stable equilibrium point here. And so wherever we start, we're going to end up there. And if we could focus here, there would be a line where if we are a little bit on this side, we make a small excursion and we arrive at the equilibrium point. If we are a bit further up, we do a large excursion and we arrive here. This is called an excitable system, and the line you cannot see, unfortunately, here, it's called a separatrix because it separates the flow into different directions. Such a system is very suitable for modeling excitable systems like neurons that when they are pushed, they make one cycle, like one action potential, and then they turn back to the equilibrium point. Another case here with 2 equilibrium points, again, you can kind of see a separatrix between them, separating the flow into 2 parts. If we are on the one side, we're going to end up on the one equilibrium point. If we are on the other side, we can lead to the other. This is a case of bistability. And so on. I hope we get a little bit the message how with dynamical systems we can represent dynamics, that is functional changes in time, in a way that corresponds to structure, dynamical structures. And we have seen a series of different objects and how they're represented in time, equilibrium points that attract the flow asymptotically, spirals of focus, as they're called, where we arrive with damped oscillation, limit cycles that are suitable for modeling oscillatory behavior, sustained oscillations, as well as from 3 dimensions and on chaotic behavior like this. Summarizing this with dictionary of terms, dynamical systems are composed by state variables that change in time. Speed of a car, in our case, what is of more interest is membrane potentials, firing rates, etc. They also comprise of parameters that remain relatively constant or short time scales. However, when they change, they take us through bifurcations, that is qualitative changes. They are described by differential equations, and they can be represented in a phase state space where the dynamics freeze, and they're represented by geometrical structures. And in these spaces, we can represent flow as vector fields, and the dynamical flow shows the direction the evolution of the system will take and the speed of it, and therefore we can form trajectories or orbits like this, describing the exact dynamics. And along these trajectories, we can have different dynamical structures like fixed points that represent equilibrium points, limit cycles for periodic behaviors, or saddles that actually send us are like crossroads. And when a system comprises mainly of saddles, we call it multi-stable. Why? Because saddles attract the flow, they slow it down in one direction, and then they let it go in some other. So these are systems where they change the whole time. They go from one state to another without ever resting, but they spend some considerable time in each one of these states. That's why we call them multi-stable, or sorry, metastable. Multi-stable, we call the systems actually where there are several equilibrium points, like the bistable system I showed you earlier, and the system can rest in either of these points. So these are kind of the terms we're going to use to describe the different population models we have in TVB. So assuming now that we have been quite experts in nonlinear dynamics, we can go a little bit through some of the models TVB comprises of. So we were exactly here. Now we understand what we mean that the dynamics of its local node here are described by nonlinear dynamical systems. And let's have a look at a few of them. One of the most frequently used is the Wilson-Cowan model. It supposes the existence of an inhibitory and excitatory population that are mutually coupled. The key theoretical idea behind it is that spike timings of neurons are random and distributed following a Poisson distribution. These are the equations. They comprise a sigmoidal function here for the activation. And depending on the parameters, they can show different dynamical regimes. For instance, this is a case where they show 2 equilibrium points for a low activity and a higher activity separated, oh, come on, by a saddle with here in the middle. However, if we change the parameters, for instance, the input coming from other brain regions, we can have actually the formation of an oscillation here, a limit cycle. And therefore, this model is suitable in general for studying oscillations in the brain. Another, and here actually is a view where you can investigate this model, and it's part of the TVB package. Here you can play, we can change the different parameters of the model and see these qualitative changes in the dynamics, both in the state space and the vector field, as you see here, and also in time series. Another model that you will see that is used in many studies, especially of resting state, it's the Wong-Wang model. This is a relatively simpler model. It can have either this state space here with only one equilibrium point, representing a low activity attractor, or it can have a high activity and a low activity attractor separated by a saddle. The state variable less here represents synaptic activity. So these are time series where only the low activity is stable, while here we have also high and low activity stable, and so we can have spontaneous jumps from one to the other. Another model that is purely phenomenological is the generic 2D oscillator. Depending on the parameters, the equations of this oscillator can take completely different forms, so we don't show them here in detail. And again, depending on the parameters, we can have something like the monostable system I showed you earlier, an excitable system that can perform one cycle only and then return to an equilibrium point, as you see here. So imagine an input here giving you a kick, making one cycle and returning to the equilibrium point. If we change the parameters, a limit cycle can also appear, in which case oscillations can be modeled. Now, in all these cases, until now, we assume that the local population dynamics are quite homogeneous. However, there is no reason we should strictly restrict ourselves in this scenario. There can be cases where the parameters of the neurons might differ. And for instance, they can form a distribution that has more than one modes. For instance, imagine neurons that receive a low input and neurons that receive a stronger input within the same neuronal population. This case is covered by this model, the Stefanescu-Girsa model, which I'm going to only show you, and it's part of TBB, and which I'm only going to show you the difference it makes with respect to the mean field representation. So on the left, we have the case where there is homogeneity in the parameters, so that the different individual neurons you see here as blue dots actually are quite well described by their mean, the mean field, which is in red. So this would be pretty much the case of all the models you saw until now. The mean field would be unimodal and fairly well describing the statistical summary of the dynamics of all individual neurons. However, here we see a case where two clusters of neurons are formed and they are oscillating. And at any case, there are two clusters, and the membership of each neuron into this cluster is changing as they're oscillating. In this case, the mean of this activity actually is meaningless. It is exactly where this model then becomes important. And it is available in TVB to model this kind of complexity in the dynamics. Okay, having gone deeper into the separate components of TVB, the structural and the functional one, we can now discuss some of the details of simulation. First of all, simulation means integration of the differential equations of the models. For integration, TVB has deterministic and stochastic algorithms. Stochastic algorithms are necessary when there is presence of noise. And TVB also has different options for noise. It can be wide Gaussian noise, or it can have time correlations, as in colored noise. So in case one chooses a stochastic algorithm, he has to also choose the kind of noise, its amplitude, etc. But the most important choice in this case is always the integration time step. A very large integration time step can make the integration unstable, and our system will explode. A very small time step will make our simulation very slow, because then the output trajectories will be approximated by many, many, many small little steps. Then one has to actually choose the measures he wants to have over the virtual brain activity. And this happens through so-called monitors that can observe the state of the brain. Now, the simplest monitor is the one that just tracks the state variables of its neuronal population. This is what we call a raw monitor. There are also versions of it that perform at the same time some kind of subsampling or temporal averaging. This would be, for instance, like measuring the LFP, the local field potentials. Then we have monitors to produce MRI Bolt through the implementation of reduced balloon Windkessel model. We have monitors to produce MEG and EEG through forward modeling, and this is where, The actual geometry of the cortical surface is very important because, if you recall, MEG and EEG depend a lot on the direction of the dipoles of the cortical columns. And also, we have monitors for stereotactic EEG, where we assume that our sensors are deep inside the brain. It's quite easy, actually, for someone to also program and develop more monitors or modify the existing ones. Now, another interesting aspect of TVB simulation is when we stimulate the brain, and therefore we perturb the TVB dynamics. TVB allows the user to easily define a stimulus that has, on the one hand, a time course. For instance, it can be a pulse, it can be a sinusoidal, whatever. And also it has a spatial structure. In other words, the user can define a spatial kernel. So he can say that I want a stimulus that will be applied at this point and then fade out as we go further away from it. And at the same time, a time course. I'm going to show you a study where applied in that way in TVB. And it is this one by Spiegler et al. What they did was to subsequently stimulate TVB at different brain regions, allow it after a while to reach an equilibrium, and perform a principal component analysis on the resulting time series and compute correlations and actually recover spatial patterns of the different networks that emerge when TVB is stimulated at different points in the brain. The result was that, taken all together, different networks appeared, and the analysis showed that they corresponded to a large degree to the resting state networks we observed in fMRI, in correlations of bold fMRI, which shows how important the space-time structure we discussed about of the brain connectivity is, because we can recover these resting state networks to a large degree. Now, I'm reaching the point where we could have a break in a while. Taking all together, the usual workflow for modeling and simulating with TVB starts with determining or loading from files the brain topology and the global connectivity, that is either the positions of brain region centers or the positions of fall triangle matrices, that if we do a surface-based simulation, and the matrices of weight and track lengths. Then we choose the functional side of TVB, that is the population model, and we can determine its parameter values. We can determine a global coupling function, how two distant regions will be interacting, in other words, for instance, a linear coupling or multiplying them or whatever. And optionally, if we are doing a surface-based simulation, the local coupling kernel, or if also we want some kind of stimulus, we have to determine also the spatiotemporal pattern of the stimulus. Then the user has to determine the measures he's going to have. If you don't select any monitors, you're going to get no output practically. So the user can select as many monitors as he wants and determine the parameters like the sampling period, etc. Then the user has to choose the degradation algorithm, and depending on whether he wants to do a stochastic or deterministic simulation, he can choose the corresponding algorithm. In the case of the stochastic, he needs also to choose the parameters of the noise. In all cases, a very important choice is the one of the degradation time step. Then you press the button and you simulate. And finally, you gather results, you analyze, plot the brain measures activity, etc. Now, further down the road, there are other things the user can do to make do some interesting science out of it. We can scan different parameter values and simulate. We can compare the activity we measure with the virtual data we generate with empirical data, and maybe then fit TVB parameters to the data, what is called model inversion. We can test hypotheses, make predictions, some of these things you're going to see at the last part of today's lecture. Now, still, TVB is a highly abstracted model of a very complicated system, the brain, and it's not straightforward to actually use it. It's not like a black box that you put together and brain function emerges. There are many common errors that can happen, like coupling functions being misused, model parameters lead to completely off-range dynamics. If the time step is not chosen correctly, then the numerical degradation will explode, will not converge. And there are even more complex problems like a conceptual confusion between structural, functional, and effective connectivity. Structural is what we get from the white matter tracks, as we discussed. Functional refers to correlations we observe in data. Effective, the connectivity is when there are really two regions interacting dynamically. And it's very often that people confuse these things, and this is very often actually when people want to model task conditions in TVB, and they think that if they just connect the different regions in the brain through the structural connectivity, and then they turn on the button, they're going to see interesting task dynamics like vision, audition, or cognition, or whatever. This will not happen, of course. And as you're going to see, until now, most applications in TVB have to do with resting state, exactly for this reason. There are additional assumptions to be done to really go from the structural connectivity we have in TVB to the specific effective connectivity that characterizes a specific task. Finally, there are two ways to use TVB. One is there is a GUI version that you can log in, you can download it, you can log in and perform all these little changes and checks, and there are many interesting views, and finally simulate, recover the data, view the data, analyze the data. But to be honest, most of the time, us doing science with TVB, we do it through scripting. Thankfully, again, there are very detailed tutorials and many examples with Jupyter notebooks, and you can all find them in the very rich documentation that you'll see on this web address. So I think we can now take a break until 7:10, I would say. And then in the remaining time, and depending on how tired you are, I'm going to take you through a few examples of doing science with TVB. I'm going to have a look also in your questions and try to respond them when we are back.
Dennis, we have answered most of the questions, but there's one request to elaborate again the Stefanescu-Giersa 3D model. It's actually not the Stefanovsky model, it's the Stefanescu. I know the names are similar, but have nothing to do with each other. So Dennis, maybe you want to say something? I have to say it's one of the most complicated models, so maybe not the best model to start. But Dennis, I would hand it over to you.
Yeah, thanks. I don't have a slide here with equations of the model. I would suggest that you really look at the paper because it's really a complicated model. I will only say a few words and you're gonna show you also a study that uses it. So let me find it. Okay. So the idea of this model, yeah, I mean, I will just show you again this video. The idea is that if among the neurons there is at least one parameter where they are differing a lot, and imagine, for instance, as I said earlier, there is an input parameter like input current, for instance, and imagine that it has a distribution across neurons with more than one mode, like a bimodal distribution, for instance. This bimodality is a kind of heterogeneity within the population of neurons, and it leads to a possible bimodality of the mean field also. In other words, the average behavior of the population is no more a good description of this population. We need to have more state variables following the different modes, as it's called, like one mode is here, another mode is here, of the dynamics. The way now this model works, it has at least 6 state variables, and so it's quite complicated one, and it does a kind of mode decomposition on the space of the parameter. In other words, it kind of, let's say, projects the neurons to their, according to their parameter value, the one that has a bimodal distribution, and like this it can describe both modes. And I guess more complicated versions could extend to even more modes than the two you see here. I cannot go into more details here. I would also have to study, to be honest, to make a better presentation of it. But you can really go through the publications here, and I hope, yeah, I hope you understand more there.
Yes, I also shared the Leon 2015 paper where there's a very short overview about this one and also the other models, maybe to start, and then you can go to the original publications.
Right, so let's continue because we are a bit out of time. Of course, the main part of the course was until here, so whatever we do from now on, it's okay. But if you want to finish it up, also we can leave some things. We're gonna post the slides anyway. So I want to take you through a few examples to show how all these things I showed you today can jointly be used and to what kind of scientific results they end up. So we started saying that EVB is the model we need to put between neuroimaging, neuroscience data, and theory. We said it's the model that connects function as oscillations in a large-scale network of neuronal populations to the observable data we have in neuroscience. And this gives us the possibility to compare the data we generate, the virtual data we generate with EVB, and the empirical data we have. And therefore, we can test hypotheses. Or when we don't have data, we can make predictions. Now, we showed also how this dynamical system we use to model brain function can undergo qualitative changes when some parameters change, like for instance the input parameter or whatever other parameter, timescales, connectivity strengths, whatever. So a usual case study, I would say, with TBB is to always try to tune the parameters of the model such that we explain something from the data. This is called model inversion. In other words, when we invert the model and we identify the parameter values that better explain a given set of data. This means, first of all, that we can have parameters that have a spatial structure along the brain. They differ, in other words. Now, there are parameters like the global coupling strength that scale all the connectivity or the speed of transmission delays, but there are also parameters that can change from one side of the brain to the other. When we do brain region simulations, these parameters can be one per brain region node. When we do surface-based simulations, they can have again some kind of spatial extent, like what we show with local coupling or with stimulus. Now let's go through some use cases, like for instance, the one of fitting empirical and simulated functional collectivity and resting state, which is a very frequent use of TBB, by the way. Here, the authors used the Wang model we saw earlier. If you remember, that model has a part of, for some parameters, it exhibits only a low activity, and for some others, it can have a high activity branch and a low activity branch. And the idea was that, okay, we have the structural connectivity from magnetic resonance data, and we also have fMRI, and we can compute empirical functional collectivity as correlations between the nodes of the brain. The idea is now, how can we tune the brain such that the TBB generates a functional connectivity that looks like the empirical one? What the authors showed was that when they tuned a parameter, the strength of the global coupling, in other words, they scaled up and down all connections of the structural connectivity, they could actually identify a region where the similarity between empirical and model functional connectivity was the best. And that was actually the region which allowed for both states, high and low activity, and an easy transition between them. In more detail, they actually vary two parameters, the global coupling strength and the speed of transmission. These two parameters affect the global synchrony of all the brain regions and their metastability. Remember, metastability is the easiness with which the brain switches from one state to another. And they showed that for a specific region of those parameters, they could actually approximate the functional connectivity of fMRI to a very large degree, as well as MEG connectivity at different bands. For instance, what they do here, they choose a specific regions as seed, and they look at the strength of correlations to all other regions for two different bands of MEG. And they do that for the empirical data, and they do that also for the model. And you can see visually how well the model represents the data. The lesson we learn from here is that structural connectivity on the one hand, and the dynamics tuned to the critical regime or metastable regime, where the brain is fluid enough to change from one state to the other, but at the same time stable enough to stay to one of these states such that we can observe it as a different network. This equilibrium between flexibility and stability that we have for a specific range of parameters is where really the brain dynamics of TBB can better represent the data. In another case, which is very similar, here the authors managed to actually capture more than the functional connectivity, additional characteristics, the functional connectivity dynamics, and there was the changes in time of functional connectivity, as well as the bimodality in the power of EEG at resting state. They did the similar thing. They varied two parameters, the global coupling and the conduction speed. And they did that by tuning the oscillators. By the way, here they used actually the Stefanescu here, it's a 3D model. And you can see that this model can exhibit dynamics like this, where there are periods of bursts and periods of quietness. And this is a kind of bimodality for power. There are moments of high power and low power. And the authors showed that for a specific, again, range of parameters, there is a high correlation between the simulated and the empirical functional connectivity for two different bands, both for tuning the system to perform delta oscillations or tuning the system to perform alpha oscillations. And what more, for the similar parameter regimes, they also managed to have a high correlation for other features of the dynamics, which is the bimodality of the power, of the EEG power, for instance, as well as the FCD dynamics, that is, the different networks that appear and the different statistics of transitions from one network to the other of the functional connectivity. All of these three measures, functional connectivity dynamics, and the bimodality of power, they all converge to a similar range of parameters for global coupling and speed. Again, this points to the specific regime for brain dynamics that are close to criticality or to metastable dynamics of an ideal equilibrium between flexibility and stability. Now, mind that this has direct clinical applications because we can now identify statistical measures that act like biomarkers. In other studies, people have compared different groups of people, like Alzheimer's patients with control, for instance, and they're trying to find biomarkers that can identify that can separate the control group and the patient group. And then this also has theoretical implications because, as we said, there is a specific equilibrium between stability and flexibility that actually leads to this narrow parameter region where we have this good fit with data. Therefore, we can have also an explanation of what changes between control and patient groups in the brain. Another more complicated case, which is where the demonstration I showed you at the beginning comes from, is this study by our colleague Michael Schirner et al. In this case, what they did is first they asked the question, how realistic is the simulated fMRI time series we get from TVP? And then they had the idea of using the EEG as an approximation of local input currents instead of a Gaussian white noise. The schema of what they actually did is they build personalized models using diffusion and MRI, as we discussed, of different patients. They place a specific population model, the reduced von Wang that has an excitatory and inhibitory population at its node of the network, and they produce artificial fMRI. However, they use also the recorded EEG as a source for noise. And they tune the parameters of the model to get a good fit for fMRI. In that manner, when the model is fit, first of all, they show that the fit is much better than any other alternative model, like a Puri noise model. In other words, this hybrid model they're using, it can fit the fMRI data much better than a series of other models like that they tried. I don't want to get into details. The good message, though, is that once they tune the brain like this, several features of actual neuroimaging data that are known in the literature can be reproduced. Several resting state networks of MRI are reproduced, and other phenomena like the relationship between the alpha phase and the spiking, or between the power of alpha and the fMRI, what you also saw at the demonstration at the beginning, the anti-correlation between them, the scaling properties of the fMRI power, functional connectivity dynamics. It seems that this kind of fit actually can reproduce many of the observations we have in neuroimaging today. Another case with direct translations to clinics is the one we discussed at the beginning about epilepsy. It's the virtual epileptic patient, and it's related to a big clinical trial that runs right now in France with 400 patients tested. The idea here is that we can use TVP in a mode similar to the stimulation we discussed earlier to actually describe the way epileptic seizure is generated at one point in the brain and transferred, spreading to other points in the brain. What you see here is the brains of patients that have SEZ channels transplanted. You see a simple seizure starting from recorded by a specific SEG contact and pretty much staying there, against another complex seizure starting from the same site, but then spreading to other channels. And you have also a simulation of the seizure by TVP. So the idea is here we can generate seizures, we can compare them with existing seizures we measure with SEG, and we can tune some parameters that will tell us where might be the problematic tissue, the one so-called epileptogenic zone. In other words, the actual tissue that is pathological and generates the seizure, and distinguish it from other parts of the network that are only propagating the seizure. So initially, we have clinical hypothesis about where is the epileptogenic zone, where is the propagation zone, and we represent it through a parameter that is like an excitability parameter. It shows how easy it is for an isolated brain region to trigger a seizure autonomously. And we can perform simulations trying different combinations of such parameters. We can actually do it systematically using a Bayesian inference method using a dedicated software stand. Don't want to go into technical details. The result of it, though, is that for every brain region, we can get eventually a distribution, a possible distribution for this excitability parameter. The higher it is, the more likely it is that that region is pathological and belongs to the epileptogenic zone. And therefore, this can direct the surgeons to actually operate this region, try to isolate it or whatever. Yeah, I don't have time to go into more details, but exactly this is what is now tried out to 400 patients in France. Here I have some other reference you can go through with applications for tumor patients, Alzheimer patients. I don't have more time for that. However, I have to say that inverting TVP is very complicated because, as we said, TVP is a big abstraction of a very complicated system. There are many things that are right now difficult to handle or can go wrong. Like, for instance, most of the error functions that we need to optimize are not differentiable, so we try it completely numerically. Usually, there are many local minima or alternative solutions that are almost equally good, and therefore, it's not easy to identify the optimum point. And this is why we generally try to optimize at the same time several features of brain functional dynamics, just like what we show in the Sirner et al. Study. And also, right now, there is no model inversion toolbox. It's something to be expected. So usually what we have to do is a very brute force grid search where we take the parameters we want to test, and we try several values of them on a grid, and we compare with empirical activity through some statistic. And eventually we get some resulting heat maps that correspond to the bifurcation diagrams, and where different qualitative behaviors of the brain are represented. And this helps us answer the question, what is qualitatively changing between one state of the brain or the other, for instance, the pathological or the healthy one. All this process requires knowledge. It's not something we can do just on a black box attitude, with a black box attitude. I have 3 minutes, so I will not go deep into the next subject, which is the last one also. The model inversion toolbox is one extension of TVP that is being worked out right now. Another one that I'm personally working with, is the multiscale simulations with TVP. I will only tell you a little bit about it. The idea is, okay, I hope I have persuaded you until now that TVB is indispensable for large-scale simulations that can directly link to virtual neuroimaging data and therefore make the connection to the reality we can measure in the brain. However, there are many spiking simulators out there that actually simulate spiking networks of neurons, spiking neurons in other words, and nest neuron are some of these simulators. The idea there is different. It's to study local mechanisms of processing, local systems and mechanisms. So are there cases where we would need a little bit of both? Well, there are scenarios like that. One scenario, for instance, is cases, what you see down here, where we want to create a realistic context for a spiking network, because usually the input coming to spiking networks is like a noisy Poisson spike train or some oscillation, but it might be the case that it is crucial for the mechanism we are studying to receive a differentiated input, either in time, in terms of oscillations, for instance, or in space, to receive different inputs from different brain regions. Sorry, this would be the case where we want to have one direction coupling from TVB to some spiking network that models a specific brain region or a specific system. There are cases where bidirectional coupling is also necessary when different modes of functioning of a specific spiking network can actually, when averaged out to update a TVB mean-field node, can actually cause bifurcations or changes to the large-scale dynamics, in which case we also have a bidirectional coupling that goes through updating a specific selected node of the brain, of the TVB, by the activity of a dedicated spiking network. In order to have this working, there are several modules to communicate activity and to transform, for instance, firing rate activity from TVB to spikes in nest, or the other way around, from spikes to create a firing rate activity to update the activity of specific TVB node. And yeah, all this is quite complicated and computationally expensive. So we're trying to have a kind of co-simulation that works actually in parallel, and to minimize the communications between the two scales, the large scale of TVB and the fine scale of nest. You can see in this, this is a graph of the performance of the tool, and it's very crucial that the parallel co-simulation is used, and also that we minimize the necessary communications between the two simulations as much as possible. And remember, this is possible because actually the connections in TVB are delayed due to the length of the white matter tracks. So actually the necessary communications between the two correspond to the minimum delay time, which in its turn depends on the lengths of the tracts in the brain. So if we simulate in parallel and if we use synchronization every minimum TVB time delay, this is actually the green line here, the whole co-simulation becomes much, much faster. And what we're also trying to achieve is a very modular interface, because we have TVB, we have some spiking simulator, nest in this case, and we have also transformer and communication modules to communicate and transform the activity from one to the other. And possibly all of this, each one of these could run on a different computational node in a cluster to allow this very large scale co-simulations to happen. Anyway, sorry, that was a little bit too fast. I just wanted you to know about it. You can learn more if you try to connect here. There is also GitHub. If you write TVB multiscale GitHub, you will also find the existing state of the code. It's really an ongoing project. It's one of the projects that tries to extend TVB to develop more its use cases. So that was all. Thank you very much for your attention. I know it was quite intense. Still, I can answer some questions if there are any.